Hey, everybody. I think it's about time to begin. I am going to start by sharing my screen. Okay, everybody. Hello. My name is Kara Wiseman. And once again, thank you so much for joining us for our second session of the Lysosomal Storage Disease Data Sharing Workshop Webinar Series Case Study number 1, Session 2, Proactive Data Sharing. My name is Kara Wiseman, and I am your co-host today with my colleague. Krista, want to introduce yourself?
Hi, I'm Chris Kazaza. I'm the Scientific Director for the Critical Path for Lysosomal Diseases, and thank you for joining us for our second session. I'm very excited to get started.
Great, thanks, Krista. We've had a great relationship here with the Critical Path for Lysosomal Storage Diseases, and we're excited to kick off this session. As I said, my name is Kara Wiseman. I'm from the Orphan Disease Center. I direct the Program of Excellence in MPS and Lysosomal Storage Diseases at the Perlman School of Medicine, the University of Pennsylvania. So let's get going. So we have a couple of housekeeping items really quickly. So one is, you've seen this before, we are taking feedback at any time. Please do fill out this feedback form to give us information about how you feel about the work going on so far, what you'd like to see more in this study, any areas we may have missed or things that we'd want to include, or just some aspects that you would like to see included in the documents that we initially create. So please, this is our website, our feedback form. If you have any questions, please let us know. Our session today and our agenda here is a real great one. We're so excited to have such excellent presenters from around the world. We're going to start off today with Laura Adang from CHOP, who's going to talk about her work with metachromatic leukodystrophy. Then we're going to go to Sean Bolton from INDPR, which is really an interesting study here talking about the International Niemann-Pick Disease Registry and their work. Then we'll be going to Özlem Golkar-Alpin, who's going to talk to us about her work in the LDRTC, and a really interesting group as well, talking about their data sharing and approaches. And then we'll go to some questions and answers and summary and wrap up with future directions. Just so you know, this time we're doing things a little bit differently, and you're going to be able to go on camera and actually ask questions directly if you would like. That will be done in the Q&A session. But if you have any questions you want to put in now, please do through the Q&A on the bottom. So a little bit more information about our workshop. What are the goals and timelines here? So we're holding these about once a month, with the exception of our last one that's a little bit later. And for these 2-hour periods, we are showing these case studies about different ways to share data. That outcome is going to be 2 different, maybe combined or separate pieces of literature that will actually work to inform both patient advocacy organizations as well as sponsors about how to share data. So this is the first time we're actually getting to case studies. I know you're as excited as I am. And so you are here in case study number 1, session number 2. Coming up, we'll have one the month after on retroactive data sharing. So how, if you started a study and you want to share afterwards and learned that this is something you'd want to do, how would you integrate those procedures inside your study in order to share? And then finally, would be looking at data sharing collaboratives. You're actually going to see a couple of the groups that we've had present throughout this time. So you'll see them more than once in more detail. So that'll be really exciting because we'll get into real in the nitty gritty of how someone sets up a data sharing collaborative. So just as a quick recap from our first one, and you'll see that you might remember some of this from our first session. And again, the video for this is now posted on our website. If you're ever interested in that or any of the presentations that have been shared, that information is available there. So basically, our real take-homes were really sharing data is much better than siloing or hoarding the data, that the NIH data sharing policy is required for compliance for any NIH-funded study, and that consent language determines the study sharing ability. We saw a lot of that information in that first session, and we're going to see a lot more for that now. So why would you do a natural history study or a registry? What can you get from that? Well, we know that biomarkers are a huge part of that, and learning about biomarkers gives us an understanding of disease progression, can help us identify patients earlier, and it has a potential for accelerated approval. So you'll see less costly trials and to get to therapies more quickly. We'll hear more about this as well later. And in addition, when you share your natural history study or registry, you're really making the most out of the investment for every patient contribution. It moves science forward, really lifting all boats. You can determine genotype-phenotype relationships, and it's a potential to be a substitute for a natural history study. A natural history study can be a substitute for a placebo control in some pivotal studies in these rare diseases. So in general, together we can help to empower the patient community to advocate for the legacy they deserve, provide sponsors the opportunity to support those communities, honor their contributions, and continue to advance science, really to find the best drug or the right drug for the right patient at the right time. So I'm just going to quickly talk about the Orphan Disease Center, again, just to make sure we can level set. And as you might recall, the Orphan Disease Center, which was founded in 2011, was meant to develop transformative therapies using platform technologies across rare diseases. And we emphasize disorders with substantial unmet need, dependent of incidence, and we strive for access to patients of all populations. We believe that being too rare or too poor should not preclude you from access to these curative therapies. And once again, just showing that the Orphan Disease Center works in the middle here in the research area as a patient advocate, but in the research side where we make patient-focused, appropriate, timely, and barrier-free therapeutic development. And we involve patients in all aspects of this work. So really we came to this because we had heard from many patients very often that they have participated in these studies, and how do they get that information out in the end? How can they use it? They want to participate in these kinds of studies, but the real focus here for natural history studies and registries are the future. So how do we leave that legacy? How do we have this information live past us? And then how do we get the data out, return and share it? And how do we analyze it for future use? So this is really what drove us to be here, to empower patients to share their data. So I'm going to go over to Krista now and let her take this part. Thank you, Krista.
Thank you, Kara. So I'm, again, Krista Casazza from the Critical Path for Lysosomal Diseases. We're one of many consortium within the Critical Path Institute. And this is a public-private partnership that, our primary goal is to pioneer actionable solutions for lysosomal disease drug development. Initially, this in our mission, we're trying to identify potential biomarkers and clinical outcomes assessments, followed by other solutions. And then with those identifications, or to make those identifications, we are creating a data repository, which is why this is so near and dear to my heart and my passion for advancing these drug development constructs through CPLD. Next slide. Just, the drug development overview, it's a multi-pronged process in which, there is standard drug development timelines within the regulatory review for different aspects. And again, for what my major focus is in biomarker identification, biomarkers come into play at every aspect of the drug development and regulatory review, which requires a significant amount of clinical evidence and data. And so that is kind of where CPATH really comes in, and CPLD in bringing the stakeholders together, and that includes industry, patient advocates, and regulatory agencies and academia to navigate the regulatory process with real-world data and enrich the literature aspects with real-world data. Next Slide, please. You can go to the next slide, sorry. So the scientific strategy is to establish that database. We call it the Rare Disease Cures Accelerator Data Analytics Platform, which aims to consolidate existing data and continue with prospective data analysis and collection to enhance our understanding and look at the current biomarkers for diagnosis, tracking disease progression, response to therapy, basically biomarker identification based on the best criteria, while at the same time looking at specific links to clinically meaningful outcomes, which we are also working on clinical outcome assessment across lysosomal diseases, specifically focusing on neuronopathic lysosomal diseases. Next slide, please. And I just wanted to bring this out. We saw this last time as well, but biomarker identification is even more essential. We know that the standard drug development timeline is different. The needs are different in lysosomal diseases or rare diseases in general. Biomarker identification then becomes even more essential because of the unique challenges associated with these diseases. In LDs and other rare diseases, biomarker identification is critical for de-risking drug development, ensuring earlier regulatory decisions, and expediting access to these therapies that we so need. Obviously there's unique challenges in the small sample size and the genetically diverse. So as Kara mentioned before, genotype phenotype relationships identified during in natural history studies or registries, as well as predictive biomarkers, help to ensure that we are getting more precise and efficient study designs. Specifically, this image also highlights where natural studies can substitute, as Kara mentioned, for placebo cases in some instances, which are often impractical and unethical in rare disease trials. So biomarkers serve as surrogate endpoints. And again, how this comes back is this all is essential. Data sharing is essential to all of these processes, and so biomarkers can help assess the different drug exposure distribution mechanisms of action in a small cohort. And so specifically, we are at CPLD really highly encouraging data sharing because it's essential nature in approval pathways and the strong clinical level of evidence that's needed. And so that's kind of where we sit in CPLD, and really along with what the different challenges that are identified across multiple stakeholders, trying to bring these together so that we can really have an efficient and effective process for drug development. Thank you.
Thank you. Now it is my absolute pleasure to introduce Dr. Laura Deng. She is an assistant professor of child neurology at the Children's Hospital of Philadelphia, or CHOP. Her work focuses on clinical trial readiness for leukodystrophies and rare diseases. Thank you so much for joining us, Laura. We look forward to your talk, and I'm going to stop sharing and you can start.
It is my pleasure. And I see in the guests as the attendees many friends, so welcome. I'm really quite pleased to be able to share our work. This is going to be slightly different than the typical structure, where I'm actually going to just show the end benefit of having collaborative data sharing agreements. So I'm going to share my PowerPoint. Does that work for everyone? I'm assuming yes, unless I hear.
Looks good. Thank you.
All right, so I'm going to talk about how within a research sharing consortium, we were able to really create an unprecedented platform for understanding an ultra rare disease. And we've started to use the same platform across multiple diseases, but the one where we really refined our procedure was for metachromatic leukodystrophy. And so Today, I'm going to focus on MLD. I do have disclosures where, in addition to NIH funding, we work closely with our industry stakeholders, Orchard and Takeda. I have lots of patient images here, all used with consent, but I think it's really important for us to remember while we're talking about sometimes dry natural history studies or data sharing agreements and such like that, the entire purpose of what we do is to help support our patients and our families. So I try and include pictures wherever I can so that our North Star is always pointed directly towards improving patient care. A little bit about myself and why I got invited to talk here is so I grew up in the South, Georgia. I got my MD PhD at UVA in infectious diseases, and along the way, accidentally fell in love with child neurology, where I did both my pediatrics and child neurology training at CHOP and Penn. Then I did a white matter fellowship for acquired demyelinating disorders, and then decided really what I wanted to be was a physician scientist focused on rare diseases. And so I pursued a master's of translational research at Penn as well. And so now I'm not a trainee anymore and haven't been for many years, but I focus on rare neurologic diseases that disrupt metabolism or biochemistry. And I work on biomarker development and validation of outcome measures, and then I partner with people in the bench lab who do therapy development, so I design the clinical trials. Very soon after I finished my white matter fellowship, I decided I needed really some dedicated time to learn how to do natural history studies correctly. And so within our research group at CHOP, we have the Global Leukodystrophy Initiative, which at that time was not RDCRN, so was not under a rare disease research consortium, but rather the Lysosomal Disease Network out of Minnesota was an RDCRN site, and they funded me for a year to really focus and hone my skills on natural history study development. And then once our RDCRN was funded, that work transitioned fully into the CHOP RDCRN. Adeline Vandiver, who leads our large consortium, has an upcoming talk through this seminar series. So the specifics of all the data sharing agreements and the collaborative network, that I'm going to defer to her talk, which is coming up, I believe, in 2 sessions. To focus on a specific disease, which has been an area of passion of mine for the last decade, is metachromatic leukodystrophy. For those of you who I see on the call, many of you are very familiar with lysosomal diseases, but for those of you where it might be a new disease, it's a autosomal recessive disorder caused by changes in aryl sulfatase A, where children cannot make sufficient enzyme and they get an accumulation of toxic sulfatides. The great news is we have wonderful diagnostic biomarkers. Newborn screening has been implemented in parts of Europe, and more than 5 cases have been identified to date. We have pilot studies that are launching and ongoing in the United States as well. We have treatments across the lifespan, particularly notable for the early onset disease, where we have approved gene therapy in the United States and multiple places in Europe as well. And we also have bone marrow transplant for late onset disease, although the outcomes from that are not as exciting as the gene therapy outcomes. We also have monitoring guidelines. So as a field over the last decade, we've made tremendous strides from a clinical standpoint of really being ready to do transformative care and early diagnostics in these children. But we have some changes or some challenges. One, so depending on where you look in the literature and who you ask, we have between 2 and 4 different subtypes: late infantile, early juvenile, late juvenile, and adult onset. And you can bend them more simply into early and late onset. But they're defined by their age at regression, right? That's historically how we divide up many of our lysosomal disorders, leukodystrophies, and even genetic disorders, right? We define them by the fulminant symptoms. So how do you define something before it's manifested, right? Our goal in MLD and for many of our disorders is to identify pre-symptomatic children and get them to transformative therapy. But if you're defining eligibility based on age at regression, but you want to prevent the regression, you get into this cycle where it's really hard. And so if you think about it in early and late subtypes, it's really important for us to be able to stratify disease risk and disease subtype in the early phases of disease. And that gets down to phenotype prediction. And so that really is my largest area of interest for MLD, is really how to do phenotype prediction and understanding that disease and mapping it prior to the regression phase. The other area of interest of our group is developing treatments across the lifespan, including how to understand that late disease course. But that's really outside the scope of what I'm going to talk about today. So again, if you break it down into early and late onset, children can either present in the toddler years with oftentimes esotropia, some questionable developmental delay, that rapidly progresses into clumsiness while walking, and then irreversible damage within the brain and the peripheral nervous system. In late onset disease, oftentimes it presents with new learning difficulties and a smoldering course that is relatively stable for years, and in some cases even decades, before there's fulminant neurologic decline. It's very easy to see the differences in these example cases when you're looking at the full course laid out neatly for you. But what if you meet a baby at birth? How do you confidently stratify into these different disease forms? It's in urgent need of us as a field. We met together as experts across multiple countries to try and to define these. But these are all, let me be clear, consensus-based guidelines based on expert opinion. They're not actually evidence-based guidelines, right? We think we know how MLD begins, but we are so biased by the children that we have met in our clinical care who are oftentimes very far advanced in their disease. And so it's really important for us to understand how MLD and these rare diseases begin even before children get diagnosed. And that's because the vast majority of children are diagnosed after the period of strongest symptoms, right? The vast majority of children are not diagnosed at birth, but are actually diagnosed years after the disease began. And so thinking of that, the lens of how we're collecting information prospectively is highly biased to a recollection bias, but also to children that have a form of disease that is less likely to be amenable to intervention. And so to address this issue, the Global Leukodystrophy Initiative's Clinical Trial Network, GLIA-CTN, has set up some core groups. And we have a biobanking core, contracts and management core, which is really our administrative core. We have a core dedicated to integrating data across the various data sources, a genomics core that reviews all of our diagnostic criteria to ensure that we can cohort our subjects appropriately based on the availability within the medical records. We have a prospective natural history outcomes core, a natural history regulatory core, which is how we collect retrospective data or existing records, and a research compliance core, right? As well as a translational research core. And so Adeline is going to talk about all these cores and the structure, but I just wanted to mention it. As part of the GLIA-CTN, we have a very large, robust research consortium. We are up to, I believe, 22 sites that are currently able to enroll and share data within our research network. Again, Adeline will talk about more of this. And all the sites shown on the side are the ones that contributed patients for our metachromatic leukodystrophy large study. And so we have several partners, including some really wonderful, strong partners across Europe that were able to share their European data. Within our natural history regulatory core, which is going to be the focus of this talk, the big challenge was how to align data sources that are so disparate, right? Most of what we were pulling were medical records from consented families. And so those medical records, depending on what hospital system you saw and where you were seen, could have profoundly different documentation practices. And so a lot of what we did was data harmonization, which is the alignment of variables across diverse structures, right? And then data curation, so looking at data quality and data missingness. And so data curation is the procedures related to data collection and organization to maximize quality. And so this, I think, sometimes the difference between a registry and a natural history study is a bit gray, but this is why I really consider this process to be a natural history study. We tried to bring the same level of rigor that you see as part of a clinical trial, with the goal of really being able to map the natural history of metachromatic leukodystrophy and other rare diseases with the same rigor you would in a prospective study or in a natural history or in a clinical trial setting. So we allow enrollment across multiple sites and multiple IRBs. We create unique study identifiers so we can map across sites. We can collect data with family permission across multiple institutions. And then we have our genomics core review and confirm the diagnosis. We set up case report forms that standardize data extraction across these diverse forms, and we create electronic databases to collect that information across the aligned data sources. And we train our staff who have to be certified in order to be able to extract and enter data the same way you would for a clinical trial. And then through this work, we can then take, say, an encounter with your pediatrician or maybe your neurologist a year before you were ever diagnosed, and we can assign retrospective function scores. And so we can map how children and adults are moving and functioning even before they had their diagnosis. And so in this case for MLD, we had 2 research coordinators extracting every single medical encounter across the duration of the medical history of all consented patients. We had a rater 1, who was me, and scoring every single encounter. There were, I think, over 1,000 encounters ultimately we scored. If the encounter was able to be scored and I assigned a score, then it went to rater 2. Rater 2 scored it. If there was consensus, rater 3, who is Adeline Vanderver, adjudicated that and signed a form that became then the final score for the encounter. If she disagreed, she looked into the medical records to see which one she agreed with. And if she agreed with neither, then the encounter was deemed unscorable. And so this is how we got a final score for hundreds of encounters. And we also did things like extracting detailed information on when developmental milestones were both acquired and lost in metachromatic leukodystrophy. The prevailing wisdom at the time we began this study was that children had a prolonged period of typical development and then all of a sudden stopped gaining skills. The classic story is the toddler that it has persistent toddling, so that ataxia or difficulty with walking, after which they had a regression phase. This didn't make sense. It's an inborn error of metabolism. Why were there not earlier developmental differences? Why was it gait that children began to have challenges? And so across our large network of both internal US sites as well as European collaborators, we identified a total of 351 total subjects who had detailed developmental histories available for us to extract from the medical records. And we could look at within the medical charts when children were sitting and walking, even from things like the pediatrician notes, or maybe they were referred to orthopedic surgery because of some gait abnormalities. We could score those same encounters and also extract if the child was walking or sitting or doing different skills at that time. And so we did this for 17 different developmental milestones and could find that a percentage of the MLD population who had early onset disease actually started to have differences in their developmental milestones as early as 6 months, which was roughly 6 months plus before they actually had regression, right? And so it's really important because this helps us to say, okay, my goal is really to treat children with the earliest form of MLD within the first few months of life, because there are already statistical differences in that population as early as 6 months. And we've used the same approach in multiple leukodystrophies at this point to really map that pre-presentation course. So in conclusion, for rare diseases, even more than any other diseases, collaboration is necessary. It would be otherwise impossible to be able to collect enough information. And it's also impossible to do many of the questions we want to answer through prospective studies because of rarity and also because of delayed diagnosis. And so real-world data is essential to fill in those gaps. In MLD, here's an example of where real-world data helped us to understand that early course of MLD and identify early markers for disease stratification, such as developmental delay was strongly associated with very early onset disease. And so hopefully this is an example where you can take from this the importance of that data harmonization and curation process. So it's taking the data and sharing data across sources, but being able to really extract and create the highest quality data that you can, so that you can start to answer very fine-tuned questions about the natural history of disease. I'm happy to pass on to the next speaker now, and I will stick around for questions at the end.
Thank you so much, Laura. That was a really great example, and dive into how you would do this retrospective analysis for the medical records. It's going to be so valuable to so many of us, because I think a lot of people don't include this kind of information. So thank you. Okay, so we will now go to our next presenter, and I'll go to Krista for her introduction.
Yeah, again, thank you, Laura. And I think it's a perfect segue into Sean Bolton, the Chief Operating Officer for the International Niemann-Pick Disease Registry. And again, it says Sean is located in the United Kingdom, but as he will indicate, that the registry, which mirrors some of the same constructs and same concepts that Laura just presented, really offers the opportunity to allow for patients and caregivers to input their data, but then Sean's just amazing application on how to really lift the Niemann-Pick community for multiple different aspects. And so I would turn it over to Sean now to hear about the work that he's done, and a very important aspect within the Niemann-Pick community.
Thanks, Krista, and thank you, Cara, as well. I'm just waiting for my, there we go, just bear with me, everybody, whilst I pull my slide deck up. And the age-old question, I hope you can all see my screen. But good morning, everybody. Good afternoon if you're based in the UK. Along with me, my name is Sean Bolton. I'm the Chief Operating Officer for the International Niemann-Pick Disease Registry, INPDR. And really, I'm going to be leaning in to the data sharing infrastructures and processes we have in place here at the INPDR to really facilitate the support of research studies into Niemann-Pick disease, some of which are natural history studies, some of which are other research projects that can be supported through the use of high quality longitudinal registry data, which we do collect here at INPDR. So firstly, a little primer on who we are. So fundamentally, the International Niemann-Pick Disease Registry, we have created and we support a global registry for the collection of high quality data for Niemann-Pick diseases, which incorporates two diseases: acid-sphingomyelinated deficiency, also known as ASMD, and Niemann-Pick disease type C. Both are lysosomal storage disorders, both caused by variations to specific genes causing issues within the lysosome of the cell, and both coming under the family branch of Niemann-Pick disease. And the registries are here to provide a platform to facilitate and support meaningful research into Niemann-Pick diseases. And the INPDR itself, we're a not-for-profit professional organization, and we're very much driven by the patient community. And the registries itself, whilst covering both acid-sphingomyelinated deficiency and NPC, we have two main sources of data collection within the registry. We have the clinical captured data, which we call the clinician-reported database. And we have the PRD, the patient-reported database, which captures patient-reported outcomes from patients living with Niemann-Pick disease and also caregivers providing care for patients living with Niemann-Pick. Going back to the patient community drive, the board of trustees, which governs the IPDR, are formed of expert clinicians, researchers, and patient representatives with the lived experience of living with Niemann-Pick disease. And we're very much guided and directed by our scientific advisory committee, who again are comprised of experts in the field of Niemann-Pick disease and lysosomal storage disorders. Very simply, our mission is to document the Niemann-Pick disease patient experience to advance research and improve patient outcomes. And we're delivering on this mission through the vision of a comprehensive international data resource specific to Niemann-Pick diseases, which informs care and supports research. A quick whistle-stop tour of the history of the registry. You can see we were started back in 2011 with the idea of a disease-specific registry for Niemann-Pick diseases, something that didn't exist at the time. And we really got our start in 2012 through the securing of funding through a European Commission grant, which allowed us to get started with creating the registry. And over the years, you can see we've hit very important milestones on our journey. And whilst we're at 2024 and here, we're very much active in 2025, as you can guess. And at this point, we have reached a recruitment of about 500 patients with Niemann-Pick disease across the world participating in the IMPDR registries, covering at least 12 countries at the moment, with a number of clinical sites participating in the clinician-reported database and 100+ patients from across the world participating in the patient-reported database. And we are working to increase that number of participating sites and patients contributing their data to the registries. So how do we collect registry data? Well, as mentioned, we have our two main sources. So the clinician-reported database, the CRD, this is our more traditional disease registry that you may be aware of and may be familiar with. This is driven very much by the clinician, and it uses the medical data of patients to inform the data entered into the registry. And the companion to that is the PRD, the patient-reported database. Now, this is a registry where patients and family members can enroll, and they start completing patient-reported outcomes with some validated questionnaires and also some disease-specific scales which are being developed for patients and family members in the Niemann-Pick disease space. We also have a mechanism in place to absorb curated data. Now, these are data sets that have been generated from separate studies or separate research initiatives, but are at risk being lost to the patient community. In session 1 of this webinar series, we dove into the importance and value of this data and how it cannot be lost and it cannot be siloed away. It needs to be available to the wider research and patient community to really maximize the value of that hard generated data. So we have this mechanism in place to absorb data sets to make it available to researchers with their questions in Niemann-Pick disease. And also important to note, and it's something we could probably go into a separate webinar session on, but we'll just touch lightly here, we have recently developed a GUID solution. This is a global unique identifier. What this means is using this GUID tool, researchers are able to link the records of patients who are participating in multiple separate studies. And also within IMPDR, this GUID tool allows patients who are participating in the clinician-reported database to match their record with their patient-reported database record, increasing the scale of the data available and giving a holistic picture of patients with Niemann-Pick disease. And for those taking part in external data sets, the ability to match data of patients participating in multiple studies is very powerful indeed, leading to higher value of that data and higher impact of having that data available. So that's something we're very excited about, and it's something we'll be launching across the community over the coming weeks and months as we've refined this GUID tool. Touching on the global footprint of the IMPDR, as mentioned earlier, we have great coverage of participation across the world. As you can see here, North America, we have a great presence. We're starting to have more presence in South America. We've recently brought on board sites in Brazil, and looking at Middle East and Europe, we have good coverage there, and also we have Australia in. So these are sites where we are working with clinicians to capture that data, or we're working with patients who are contributing data to the patient-reported database. But then we also have countries where we are actively working to increase the presence of IMPDR, to increase the opportunity for patients to take part and contribute data. Now getting a little more under the hood and looking at the data sharing infrastructure that we have developed here in IMPDR. Because going back to the mission of the registry, we really are here to support and advance research in pneumothorax diseases. And we have a number of infrastructure elements in place to really support that proactive data sharing using registry data. And first and foremost, the core process we have in place to support data sharing, and it's something I'll get into much more detail in a few slides' time, is the data access request process. This is a process that allows third-party external applicants to submit research proposals to the IMPDR for review and consideration, and for the registry data to be provided to support the analysis and hopefully answering of that research proposal. And that's something I'll get into in a few slides' time. A key infrastructure component is the consent-based sharing model. Now, this isn't registry data that we have. This isn't IMPDR's data. The data that is held in the registry is patient data, and we believe it belongs to the patient. And what this means is we make sure that patients have the opportunity to dictate how their data is used. So within our IRB approved materials, such as our patient information sheet and our informed consent forms, we lay bare what can happen with a patient's data, how it's shared, and how it can be used to support research projects. And in the next slide, you'll see a screenshot of what those consent forms and information sheets look like. And you can see the language that we use, but also the data sharing options that we make available to patients in the consent form, so they can clearly state how they wish their data to be used. And we think this is a fundamental element of that proactive data sharing. If we can be confident that we can capture the wishes of patients and how they wish their data to be used, then we can be confident that we are serving the patient community by using the data in the way that they wish it to be used. Another key component of our infrastructure is having a secure platform to host the analysis of registry data to support those data access requests. We collaborate with the University of Dundee, and we utilize their Safe Haven platform. Now, this is a virtual environment where we can upload registry data sets, and we can handle secure access of those third-party applicants to that registry data. What that means is we're not providing unrestricted access to registry data into the registry system. We're only uploading data sets that are required to answer the research questions that the data applicants put forward. And in this controlled environment, we can regulate who can access it, how they can access it, and what kind of analysis they can undertake to make sure that whoever is accessing the registry data in that secure, safe haven environment is only undertaking the kind of analysis that they have approval to do. Complementing this collaboration with Dundee, we also have a collaboration in place with the Critical Path Institute and with Krista, who's been fantastic in really supporting us in developing this part of our infrastructure. And what this collaboration with CPATH allows us to do, again, it's another secure environment where we can host registry data to support these research questions, but also using CPATH's capacity for analysis and expertise in biostats and other areas required to undertake analysis. Data access request applicants, if they don't have the capacity to undertake analysis, they can lean in to CPATH's resources and capacity to really answer those questions. From a agreement level of how the data is used, we have a number of agreements in place that we put in with our various partners and collaborators, again, to govern how that data is being used. For example, with our clinical sites who are taking part in the clinician-reported database, we have data sharing agreements, again, laying bare how data is being transferred from the clinical site to the registry system, and then how data in the registry can be used, again, to support research questions. For those analysis platforms that we work with, University of Dundee and Critical Path, we have data processing agreements, again, governing how the data on those platforms are used and how those organizations can use registry data. Again, we have the rules of engagement laid clear of how data sharing happens. And also for data request applicants, we have data usage agreements, again, just putting in rules of engagement of how the data is being used to answer those research questions. And finally, we have a very robust system in place to work with pharmaceutical industry partners coming under data access requests. There is a high degree of sensitivity in the community, and understandably so, about how organizations can work with pharmaceutical companies in providing data. So again, this is something that we have given much consideration and much consultation with the patient community, and we have a model in place to allow us to work with industry to answer those research questions that they have, but to give assurances to the patient community that we are working in an appropriate and in transparent manner with these companies to maintain the faith of patients who support the IPDR. As mentioned earlier, this is a screenshot of our consent form and our patient information sheet, again, talking about how the registry can use data to support research proposals as they come in. And as you can see on the left-hand side of your screen, this is the language from the information sheet, clarifying how we could be using registry data. And on the right-hand side, you can see a screenshot of how patients can indicate their data sharing preferences within the consent form. And just to note, these documents have received IRB approval, so we are confident that they are of a high ethical standard and that they are written in a way which is clear to patients and allows them to really give informed consent about how their data is being used and also how they are participating in the registries. So the data access request process, as mentioned, this is really our key infrastructure to allow applicants to use registry data to answer their questions. And it's a comprehensive research process. It's a multi-step process allowing us at various stages to check the credentials of the application coming in, making sure we have sufficient data quality and volume to answer the question, allow the research proposal to undergo appropriate scientific oversight, and allow for a decision to be made of how that data is approved to be used to answer the research question. And these applicants can come from any background. They can be clinicians, researchers, industry partners. They can also be patient organizations or from any other backgrounds such as regulators. Again, we use our collaborations with Dundee and their Safe Haven or CPATH with their research environments to allow that secure sharing of registry data. And as mentioned earlier, there's no unrestricted third-party access to the registry data. It all is held within a secure environment. And I'm not going to focus too much on the benefits. Again, going into webinar series 1, the challenges in generating high quality data in lysosomal storage disorders and in rare diseases across the board. Registries aim to reduce those challenges and minimize them as much as possible. And our data access request process is designed to maximize utility of registry data whilst minimizing potential barriers to applicants in using registry data. We ask for applicants to complete a template request form and to indicate which variables that they are looking to use. We are assessing the background of the study and the research question that the applicant is coming with to the registry. We're interrogating the variables that they're asking for, and we're looking to understand what is the output expected from this research question. We look at the governance process and if we need any further approvals, such as local IRB approval for that specific application. We ask applicants to confirm which analysis platform they would like to be using. We ask them to agree to a publication policy to acknowledge contribution of registry investigators. And when it comes to funding, if there is a request from a pharmaceutical company or from an applicant who has funding specifically to answer this question, we will seek funding from that. However, if there's an applicant who has no specific funding in place, then we will support that data access request for the advancement of science and to improve the outcomes of patients living with Niemann-Pick disease. In a nutshell, and at a high-level summary, we start from left to right. So in the first instance, the data access request is submitted, again, formed of the proposal form and the variable selection form. These forms will be used as part of a feasibility review, again, looking at the data availability to support this research proposal. If there is sufficient data available, the request will be escalated to our scientific advisory committee, who will review the request, and they'll generate an opinion of whether to support or reject the proposal. This will then go on to our board of trustees, who make final decision about how that data is shared, if it's an approval of the request or not. For approved requests, we go on to the dataset preparation and anonymization. So again, there's a number of steps here that we undertake to minimize the risk of identifying participants. We look to share only anonymized datasets with applicants. Once we have this dataset available, it's uploaded to one of the secure platforms, and then the analysis is undertaken by the applicant. So I just want to give you a very quick example of some of the studies we've supported so far through this process and that have used registry data. So one of the first requests we received was from researchers at Great Ormond Street in London, and this was looking at early and late infantile Niemann-Pick disease to better understand the disease progression and to have a forward look as to how we can use that information to produce better design of clinical trials to accommodate these pediatric populations. And this request resulted in a number of posters being accepted at multiple conferences in the lysosomal storage space and also in the rare disease area. A study we have currently ongoing at the moment is looking at seizure burden in Niemann-Pick disease type C, led by a clinician out of Central Manchester Hospital University. And this is looking at the incidence of seizure in Niemann-Pick disease type C and trying to see if there's any correlates with other features of Niemann-Pick disease type C, which may help inform some of the direction and the prognosis of patients. And this analysis is being undertaken at the moment with our partners at CPATH to support Dr. Donald in undertaking the analysis. And finally, a piece that was generated from IMPDR itself, we undertook a characterization of patients with NPC in using the registry data and trying to understand a bit more about the demographics of NPC patients in the registry, diagnostics, other signs and symptoms, and looking at treatments that patients have experienced. And this was an open access publication in Orphanet Journal of Rare Diseases about 3 years ago now. So again, just a brief glimpse at some of the studies that can be supported using registry data and which have gone through our data access request process. So to leave things on a quick and dirty top 3 tips from IMPDR about how sponsors looking to create their own registries or natural history studies and are looking at proactive data sharing, I think this has to be something that has to be thought of from day one, possibly earlier, when you're designing your study or designing your organization to run this registry or natural history study. You need to look at the governance structure in place to make sure that you can facilitate data sharing from early as possible. Look at your patient-facing material and see if the language in there is such that it really informs patients of how their data can be used and how they can specify how their data is used. Look at your internal SOPs and processes to see if you have the capacity and the capability at this time to support data sharing. Look at some partnerships that can facilitate that data sharing, for example, with CPATH for ourselves and Dundee Safe Haven environment. The sustainability of data sharing is key. It's a costly and time-intensive process. So identifying diverse funding streams and grant opportunities to support not only the delivery of your registry and your natural history study, but to support the proactive data sharing element of your project is key. And finally, from me, looking at internal staff capacity and capability, the processes that we have in place are fairly complex and again, time intensive. So making sure that you have a clear process in place from day one and you have the expertise within your organization, or having a strategic collaboration with an external party who can fulfill those features and those functions is super important. I think this list could be another 5 or 6 slides personally, but from my side, this is the top 3 recommendations I would give in terms of having that proactive data sharing. And I'm happy to talk more in the Q&A section. And thank you all so much for listening to me this afternoon. Thank you.
Thank you so much, Sean. That was really excellent. Yes, these presentations really do pair off each other really well. And thank you so much for giving a review there, talking about the way that you guys approach data sharing. So we will have our last presenter, which Krista will be presenting or introducing. And I will share this. Second, please.
Thanks, Sean. Thanks, Kara. Yeah, I want to introduce Dr. Aslam Gopar-Alpan. She is the founder and the president of the LDRTC. This is a nonprofit organization focused on lysosomal diseases and other rare diseases. She established LDRTC in 2013 and really has, with the vision of quality care, but really the passion and the effort is so well recognized. And I'm very, very excited to have Dr. Gopar-Alpan here. She has a medical degree from Marmara University in Turkey and graduated at the top of her class and chief resident in pediatrics at SUNY Stony Brook. She has a long history of vast experience, and her commitment to the future of healthcare in lysosomal diseases is evident. And very happy to introduce you, Aslam, for your presentation.
Thank you, Krista, for this very kind invitation first, and also the presentation introducing me. And I am really delighted to follow these wonderful 2 speakers. So today what I want to do is I want a little bit actually, have the gears think about how we can set up not only data sharing. I mean, we're mainly discussing the clinical data right now, which is in dire obvious need. Also set up a biorepository. So this is what we have been trying to do for the past decade at LDRTC. I'm going to give you to the attendees a background, what we are and who we are. The reason is we are truly a unique organization. So I'm just going to start hopefully sharing my screen right now. Is everybody seeing my screen?
Yes, you're good.
All right. Okay. So I want to actually have a title called Data Without Borders. The reason is we truly believe in the notion that the only true owner of the data is the patient or the subject. So the data doesn't belong to us. Data belongs to them. And if we are lucky enough to share with the other researchers, I think we are very privileged. So who we are a freestanding research organization. We mainly do clinical research. At one time, we conduct close to 30, 40 clinical trials that include several registries from phase 1 to phase 4. We currently actually run about 6 gene therapy trials, other small molecules, enzyme replacement therapy. We focus on lysosomal disorders, basically on sphingolipids disorders. So how the organization is set up, I'll give you a background. I mean, I worked at the NIH for, over 15, 16 years. And the reason why I left NIH was partially that is during the shortage of enzyme treatment. for the patients, starting with Gaucher, Fabry, so on and so forth. And the patients really get disfranchised during this time, and not only with their care, but also, what we can provide as clinical trials. So, and the other thing that hampers the patients with, or hampers patient care for rare disorders is fractured care. I mean, usually in large academic centers, we are truly indebted to centers of excellence. But what happens is the patient, actually, the ones that I take care of, like if you give an example for patients with Fabry disease, their major organ involvement includes the heart, the brain, peripheral nervous system, and also kidneys, obviously. So they are the prescription is provided usually by a geneticist, rarely by a cardiologist or a nephrologist, so who takes care of their enzyme replacement therapy. But on the other hand, these patients need care for all the other organs that are involved and need to be followed, and they need to be put on transplant list, this and that. So basically, they end up having a list of 10, 15 different items to follow, physicians to see. So basically, truly, this has never been followed. So patients actually follow with one or two subspecialists. They may have, this day Fabry Day, they may have 5 different specialists, but truly that's like they don't have an echo that day, the MRIs are delayed, so on and so forth. So basically, fractured care is the primary problem for patients with rare disorders, lysosomal diseases. So we basically want to actually engulf or incorporate all the different, meaning the clinical care, clinical trial, and also research, also the procedures that includes like echocardiograms, physical therapy evaluations, pulmonary function tests. The only thing that we cannot do is the MRIs. And we do small procedures like muscle biopsies, skin biopsies, so on and so forth in the premises. So basically, we are very adamant about data sharing, and the reason is we have been collecting these, what is called so far is the curated data, or very well phenotype patients for over a decade that visit to us every 6 months or more often in occasional instances. And we have very detailed phenotyping, including genotyping and other collection of the biospecimens. And why I am insisting on biospecimens, as we discussed for both MLD and also Niemann-Pick diseases. And when you come with the biomarker, the fluid biomarkers, which are non-invasive biomarkers that we're talking about, you need to have the correct phenotype given that time with the progression of the disease. So it would be very important to know when the patients starting falling off their curve developmentally for the MLD, and what happens, for example, when they receive the gene therapy. Is that right? We need to compare before and after. This is very important. And at that time, you need to have a biorepository that is attached to a clinical data set. So we are different than academia, like CHOP, and we are similar in a way to CHOP too, in a clinical environment. So the similarities, so basically we are completely research focused. We started from the clinical trials. We also actually have educational efforts, and we do some sort of advocacy too. We took the advocacy efforts for neuronopathic Gaucher disease. We are having our second patient meeting this year that we're going to be focusing on the infants and small children and their parents. But we are different too. We are not university-based. We are freestanding. We use central IRB, which is actually becomes very important when you are planning to start a trial, when the time is essence. So we are not bound with the, with big academia IRBs, and also the budget negotiation, the contracts actually run more swiftly. We on the downside of it, are actually the funding is very limited. So we are basically operate on grant-funded studies, And we do have donations from the patients and similar entities, but we try to use them for patients only. So we are not really using them for our day-to-day operations. So what we are is we are truly patient-centric. So we do anything and everything just for the patients. Obviously, every center is doing the same thing. What I mean here is for simple projects that have a beginning, middle, and end, and also can be used for purposes like drug development, which drug to choose, so on and so forth. And we are unique in a way that we are a translational research facility. So when I worked at the NIH, if NIH was built as the clinical center as a little cross or a T shape, so one side was lab, one side is the clinic, and the others are the labs. So patients the building was designed for the patient to be wheeled in from the lab to the clinic, to the lab. So obviously we don't wheel any patients to the lab, but there is a small biomarker research lab that we do molecular, more cell biology, and we recently acquired a mass spec LC-MS to do lipid and protein-based biomarkers. And we provide treatment on one side too. So this is a little, patient care facility. And this is the research laboratory. They work hand in hand. This is the clinical research team. And these are this is the most staffed team that we have. We actually have about close to 8 or 9 individuals that work on clinical trials only. So basically, the similarities with academia for data sharing is obviously we follow IRB, HIPAA, and patient privacy regulations. And data transparency, we are very much actually put an emphasis on the transparency of the data. And obviously all data is de-identified. And when we share with the other researchers, and when the studies are done, for example, from the translational lab or the clinical unit to the translational lab, also the data is de-identified, that every patient is given a number, and actually the key is kept in a locked cabinet or under the password in a computer. So similarly, in the clinical research, the patient, there are only initials, there are no names. So basically these 3 different units for the patient privacy, they don't intersect except for the numbers and the patient initials. So we do collaborate we have done with multi-institutional studies, and we have done partnerships, obviously, to publish, and all of them actually have been published. And as I said, the funding is grant-based. We don't have any NIH grants yet. We do have NIH fellows that come and rotate with us for teaching purposes. And all our publications are peer-reviewed, and we mostly put emphasis as an open access, as our primary goal is to share knowledge with the world, honestly. So one thing that we put emphasis on, if you Google me, I have participated in the CME or other conferences that we actually have an enduring part, almost a year or more. And how the attendance to these conferences is about limited to 150. They're very small conferences, but when we put on the web, usually the attendee list can go up to 3,000. And we have attendees from all over the world, including Nepal, Tibet, Africa, other small countries that normally they cannot attend these conferences. And we put also emphasis on sharing knowledge, including collaborating with multiple countries in the other instances, as the presenters put emphasis on. So basically, we are focusing on lysosomal disorders, including, most emphasis is paid on the sphingolipid disorders. The reason is they are the most common. And our primary goal is patient-centric. In the meantime, the patients are also provided service as they are participating in research endeavors. And the data access is structured, or as I am speaking, is how we collaborate with other researchers is basically through our repository, and the data access is structured under an MTA, which is quite simple. Basically, they cannot sell, they cannot use for other purposes, and everything needs to be transparent and published in that MTA. So our focus is on Gaucher-Fabry and Pompe diseases, MPSs. So we have started a recent trial on use of amroxal in MPS 3. We have made the drug produce and actually have an IND and pass through USDA. So basically we can actually design a trial and run a trial. This was through the Sanfilippo team Sanfilippo, their advocacy group, and our collaboration with them. So we do from phase 1 to phase 4, we run multiple registries. So actually I have one question about the registries too. In one instance, we were running about 4 different registries for one single disorder. So we had a workshop in past years, asking the question whether we are able to get all these registries together like they have been done for MLD or Niemann-Pick, and where we can collaborate with some data, because when you run a very large registry, so we're talking about like for Gaucher disease, more than 5, 6, 7,000 patients, the data is inadvertently becomes unreliable. So basically you have the demographics correct and you can't really rely on whether the genotypes is correct, or you can have a genotype, the most, for example, for Gaucher disease, alpha-4P is the most common genotype for neuronopathic. And they sometimes they don't really list the other allele for the neuronopathic Gaucher disease. So basically, you really don't know whether this is a type 1 patient who is severe with an alpha-4P allele, or a type 2 patient with a really another severe allele that actually progresses very rapidly. So basically that is our scope includes tracking the progression for the disease and for future collaborative work. So the data we collect and we have shared that include the clinical records, imaging. So we have access, and in our repository we have the MRI imaging, the organ volumes, brain MRIs to a lesser extent, lab results, genotypes. So the genotype or the genetic data can only be accessed and be shared by under a different kind of consent form. So that is not within the umbrella protocol that we are collecting the biorepositories. So in our biorepository, we have been collecting blood that includes serum plasma and then frozen PBMC and DNA, RNA that is being collected for over a decade now. They are kept to -150. That is now actually we are purchasing nitrogen tanks. The reason is for future actually use and also for the backup. And all these could be shared per the MTA under IRB reviewed or IRB protocols. We do not actually participate with any data sharing without a certain study that is under the standards of good clinical practices. So in our consenting process, I said the genetics and genomic studies can only be done by a separate consent form, but this is our biorepository collection protocol, which is, we call it umbrella protocol. It is disease agnostic. Basically, it is any patient that comes to the center. We have been collecting close to actually 700 to 800 samples. Patient patient data sets that include samples, that includes both lysosomal and other disorders that include rare immune deficiencies and some other rare disorders that we actually became a part of the identification of that disorder. Obviously they said the consent process can be arduous. We try to keep it very, very simple. Our consent is 6 to 7 pages, and we use Verb as a Western institutional IRB as our central IRB and a local IRB for this purpose. And there's a certain language that needs to be incorporated. So mostly it is that language we share. We say that we are sharing your samples unidentified, or we would be sharing with other researchers under other research protocols, and they are made aware for that. We also have skin biopsies for cell biology studies, fibroblast cultures. We have been collecting urine samples. We had published something on urine kidney epithelial cells. And I am pleasantly surprised to see there is a new publication coming. Another group has been using these cells for genomic studies. So basically we publish them as a mean of drug screening studies. So we also collect leftover tissues from other clinical procedures, and the patients are aware, any kidney biopsy, muscle biopsy, so on and so forth. And we have few donated organs by the patients or the patient parents. And all the specimens, as I said, are de-identified and securely stored for future research purposes. This is a brief actually screenshot about the patient's information about what happens to their samples and how the data is stored. So basically they are informed that the samples are collected for research and for research purposes only, and will not be sold or distributed for other purposes. So there was actually with the Niemann-Pick, the collaboration with the industry. So we really don't give any samples to anyone, including industry, except for if there is a collaborative project that is going on with the industry, we do on a grant basis. So basically these samples are only shared when we know that there is a project going on and with the purpose, and we can provide or collect samples for industry with an IRB reviewed and IRB approved project. So basically the patients give permission for collection of the tissues removed during non-research clinical protocols or procedures, and they can say yay or nay. If it is yay, nay, then unfortunately leftovers will be not stored, but they will be thrown away. So basically the legal framework as an MTA is obviously we provide the ownership. However, with the confidentiality, they're marked as confidential and they're only restricted use under that MTA. And we are compliant with HIPAA, GINA, and other local Virginia third-party laws. And so for collaboration, this is our part of our clinical research team. Each coordinator is responsible from a protocol where we actually collect and share data with other researchers. So we keep our Excel sheets, whom we share the samples, how many samples were sent, the dates, and also we follow the process of the project, and also for future publication endeavors. So we have a legal and IP team. So if there is a joint project, we try to have an IP before we pursue the project if there is a patentable or intellectual property. And so basically all the researchers that goes under this MTA, they are bound by the confidentiality. Results are mutually shared. So if we are collecting the clinical data, we will share with the researcher. if they are collecting the research data, then we have routine meetings at certain intervals that we actually share the data each way and see how the project is progressing. The reason is these are rare disorders, and this the biorepository has been collecting with really endeavor, both financially and also the staff-wise. So we really do not want to invest in any project that will not have a beginning, middle, and end, meaning that we will not share any sample that we think that the process is not in place for a future collaboration or any publication or any real work that really pertains to the patient. So it is exclusively non-commercial. There is no external transfer, and we can breach if our terms are breached. We can terminate the collaboration anytime during the project. So how we are envisioning to make this actually be available for other researchers than the ones that we collaborate with under the MTA. So we will actually have the data collected under the NCATS eligible radar for broad access. And the procedure is de-identify and obviously at some point submit the radar and request via portal that can be reviewed by the LDRTC for the correctness of the data. And the cost is there is no fee for sharing the data, including the biorepository specimens. It is done under a funded through a grant. So basically the MTA terms, they're nonprofit, academic status under DUI and DUA and restricted to services that is in the MTA. So the researcher cannot get the biospecimen and use for another study. So we don't charge any fee and or a nominal fee under a grant for the shipping and handling of the specimen because they need to be towed, and then we need to have FedEx or other courier services that requires. Actually, we were sharing samples with Canada, and our dry ice expenses was really crazy. It was more than thousands of dollars because the dry ice had to be replaced at the customs. So that the funding will be used for this kind of purposes, but not for the biosamples. So we require an IRB review or IRB approval and the LSD research focus for the repository access. So we don't want these samples to be used as code-by-code controls for any other disorder. So we have actually completed multiple projects so far, and we published on all of them. So for Fabry disease, for example, we have shared our samples with the Jackson Labs. If one can remember, the NKTI, the receptor is the GLA. And that was one publication, and researcher was including as actually they were very interested to look into that. We found other things, not NKTIs though, because they're very rare cells. For Gaucher disease, we have collaborated with NYU for immune exploration of inflammatory markers in Gaucher disease with their treatment status. We have done a 3D bone model in Gaucher disease with Penn State University Engineering. So we also try to actually incorporate researchers that are usually not the players in the field. That is like we try to incorporate like engineers. We will have hopefully a project with Argentina and a university in Texas for drug screening with the micro nanotechnology. So hopefully, that's the engineering department. And we have completed a pregnancy study with Duke University, and also we actually had 4 or 5 researchers identified any disorder that is the infantile spastic paraplegia gene with the UBAP mutation. So we have contributed the clinical data for that. All right, so basically, how we would enable data sharing in new trials for the future. So basically, our goal is to facilitate sharing with researchers for the trials. We still want to keep the similar ethical aspects of the sharing. However, we want to actually expand the biorepository storage unit that we are incorporating nitrogen tanks for the long-term specimen storage. So basically sustaining the data. So one is obviously the cost. Like currently the cost is funded by individual grants for each project or from our own budget. So we do not accept any funding from industry to collect data. And we want to actually have this to radar accessible for long-term access so we can collaborate with NCATs or like clinical CPAT here, so we can share the data that is collected. And also the succession is not person dependent. So we have seen the examples of this in multiple disorders. Like one researcher get interested with disorder and then collects samples for over decades, and that the researcher retires, and we really don't know what happens to those samples, or a catastrophe happens like a storm, and since the data and the biorepository is not primarily belonging to the other researcher that inherits that repository, unfortunately we have seen that the many specimens perishing, unfortunately, so far. So I would like to thank everyone that is presenting here, and especially Krista and Kara, and hopefully that is CPAT will provide a venue for the data sharing under different projects, but also as a repository for the biospecimens at some point. Thank you.
Thank you so much. Gosh, we have such a great overview of different ways that people do data sharing now, and this added so much to our discussion. Thank you very much, Olsum. Now we are going to go to our Q&A. Going to just share our current panel here. So this is where this is the panel that we will now be working with. And I'm going to invite all of our speakers back on. Hopefully Laura can join us as well. Thank you. Oh, great. Excellent. So we've had some really amazing presentations here that really go over so many different aspects. And we are going to welcome people not just to, here, I'm going to stop sharing here just so we can bring people back into the conversation from the Q&A. Why don't we start with our actual Q&A here? I think that you guys can read these as well. But let's start with our most recent one. Teresa Berger asks, and unless you would like to speak, Teresa, yourself? If not, I'll just speak for you. So data requests from researchers are common. How are data requests handled when the requests come from the participant and or the disease community itself?
So I think I'm going to leave the disease community. So basically we are talking about advocacy organizations and or patients here. So that is a little bit more tricky than sharing with the researcher. The reason is, you know, if a patient has the disease, then or an advocacy organization for rare disorders like Niemann-Pick, that the patient or subject or the information could be identifiable. So in that case, what we do is we try to change either the birth date, the order of the events, not really impact the whole clinical picture, but make it very unidentifiable to the patient community itself. And the other thing that we can do for this instance is if it's going to be just a biorepository project that we have done with Howard University. So what we have done is we collected about 5,000 samples, and we for that attempt to identify treatable forms of lysosomal disorders in the minority populations. But then what we have done is we made them completely unidentifiable. So we were only had the gender of the subject and the age that the subject was when the data was collected. So nothing. So this was an IRB exempt protocol. IRB reviewed and exempted. So the other option could be that an IRB reviewed but exempted study where everything is blinded and they only get the sample or data that is needed. So about the other project, as I said, it is what we do is usually collaborative projects. So we don't really just provide X data like the imaging or the developmental milestones and so forth for a researcher for the sake of just providing it. So there needs to be a sound research question that is going to be a collaborative effort than we just being the provider of the data. I hope that answered the question for Teresa.
Thank you for that answer. I just wanted to briefly say, one of the things that we found is a lot of the patient families and patient advocacy groups say, please share my data with everybody. Please spread the word. A lot of them don't feel that there's a need for, keeping things so secretive that they can't share information with each other and support each other. And I think that's an issue. Regulatory wise, we feel like everything has to be protected and kept secret. But the families really don't necessarily want that, depending on the families and the advocacy groups. Can you speak to that a little bit?
Yes, absolutely, because we do have these patient meetings too, and we, with their approval, we try to put them in the web so that it becomes an example. But then we have to get their consent for sharing their images or image likeness or their views and this and that. So basically, yes, but for whatever you're saying, particularly for their information, I mean, we try to make books. We produce a book on Gaucher disease, and we said it's the patients or my journey, and we actually had the real stories with the patients' images. We kind of made them altered into pictures, so they were actually figures and the book pictures or cartoons rather than the real likeness to the patient to change it. So basically, when you are dealing with HIPAA and is in our shoes and patient privacy and also the future of sharing. So basically, when we are talking about an adult patient or a child is very different too. So now there is the whole question in the web is whether the parents actually have all the right to share every single minute of their children. Okay, so basically this becomes a big privacy issue, and whether these children would be agreeing when they are 30 something for their images when they were at 10. So basically there are many ethical concerns and questions to share this kind of data as a researcher for us. I mean, when we publish, we explicitly get consent forms for their images for that particular publication. And I do have patient videos and patient narratives that I use for teaching purposes. And we actually have consented the patients to use them with other researchers with the idea that they are going to be only used for teaching purposes only. So industry occasionally comes to us and asks, for me to prepare presentations or to share with the public. So that becomes an issue too, because these videos are obtained, belong to a patient, to a person, and they are not actually for profit use, even though industry has no intention or might not have the intention to use for a profit purpose in that instance for teaching purposes, but still industry is a profit organization. So that becomes multiple issues and concerns, for especially these rare diseases where the patients the number wise are limited. And also, while we want to spread the word about their journey, we are limited with the respect to their privacy and the laws that actually apply to that.
Thank you very much.
Thank you. Sean, would you like to speak to that as well? And then we'll hear from Laura. I think this is important. Oh, sorry, Laura, I didn't see your hand was up.
Sorry, thank you. So yeah, absolutely. So it's striking that balance, Teresa, isn't it? It's really the wishes of the patient community, and we hear that a lot. You know, use my data, make it valuable, maximize what it can contribute to the knowledge pool. However, we're trying to balance that off with the ethical constraints and also data privacy regulation as well. You know, from the European standpoint, we are an international organization, so we work across many jurisdictions, but general data protection regulation, GDPR, really does constrain what we can do and how we can set up data. So we're striking that balance between maximizing patient wishes of use of data versus the regulation that we absolutely have to abide by. And I think the model that we've struck on, and I think we've you know, getting to this, there isn't one model that fits all. It's working on a per case, per organization, per purpose basis. But what works for us is I guess the design of our consent forms and information sheets really does put the tools in the patient's hand of how their data is shared, and that gives us the balance of what we need to do to be compliant with ethical regulation and GDPR and data regulation. And from our organization perspective, we have a data protection officer in-house. Now, this is an individual who really works with the interest of the patient at heart and really looks at these compliance questions and these ethical questions and looks at them through the lens of, okay, what is protecting patients? What is protecting their special category data, genetic information, and other sensitive information? So making sure that we are giving patients and participants the right information to really go through that informed consent process, so they're coming out with the right decision for them at the end of that, and they're indicating what works for them as an individual.
Thank you. That's awesome.
Thank you. Go ahead, Laura.
Yeah, I was just going to add in. So, as the academic representative here, I think our lens is even sometimes a little bit more challenging because we are also the clinicians caring for these families. And so I think we have an extra level of protection that we have. And I will say the consent process within an academic center, we do not get that same universal use my data however you want. I think it is the lens of the families that are reaching out to your groups that say everything to everyone. But really, from the clinical side, we do see a lot more restriction coming from our families because we're the first research place that a lot of our families are encountering. And so I think I want to be mindful of, depending on the population that we're seeing, we may actually see very different views on data privacy. And we see the full range of families that do want to be more restrictive with how their data is used. They don't want it shared with the NIH. They don't want it shared externally. And so I think it's being flexible and mindful, but also able to track those differences. I think that that's a big change. I think in my professional career, one of the most exciting changes has been recognizing and involving all the stakeholders in research and really bringing in our patient advocacy groups, our industry stakeholders, and learning from each other so that we're not reinventing the wheel, and we're trying to make sure that the information that we get from our work has the highest impact. And so to that end, we have built in a structure within our rare disease consortium that is led by the patient advocacy groups. We have, oh goodness, I think 40 to 50 PAGs that are part of our RDCRN. So leukodystrophies are a very diverse group of over 200 genetic disorders. And so we have regular meetings with our PAGs, and then have established new processes to allow bidirectional data and information flow. Because I think that you're right, our stakeholders, the most important one are our family advocates. And so we need to make sure we're both doing research that is important to them, but also having the ability to help support the work they want. And if it's okay, I'm going to throw it to Melody Kaiser, who leads our PAG group as part of the GLIA-CTN. Melody, are you okay with coming on?
Hi, Laura. Yes, thank you. I'm here. Let me see if it'll let me. I don't think it's gonna let me put my camera on, but I'm here if you can hear me.
Yes.
Wonderful. Well, thank you so much for passing it over to me. This has been an incredibly informative talk, and I'm so grateful to everybody who's on this call. I have to say this is a major priority for the leukodystrophy PAGs. We keep hearing over and over that that families want to contribute, they want to help, they want to give their data and share whatever they can to advance research. Oftentimes knowing that it's not going to help their own child, but feeling like it's some way part of the legacy. And it can be so frustrating when there are roadblocks to data sharing or advancing the research. So I'm so grateful for this group coming together. How can I contribute to the conversation? What's the most useful way I can share?
Well, can I say that you guys met last year and came up with a list of requests for us
We
of
did.
ways we could be better partners in this space? And to me, that was a transformational, like really beautiful, well thought out letter that we received of these are our points as PAGs. These are how you can better collaborate. And we have spent the last year trying to adapt our program and our research approaches to meet as many of those things. I mean, I think we come in with a very paternalistic view, or in my case, maternalistic view as researchers, where we're trying to guess or anticipate what families want and need out of their research. And oftentimes we're right, and then oftentimes we're not. And so one of the things that we came up with was a public facing website, and that oftentimes our PAGs don't want or need the 8 hours per patient detailed natural history study. They want, how many patients do you have consented? What are their age ranges? What genotypes do you see commonly? So that they can go with that information to other families, to stakeholders, to potential investors, right? They can drum up industry support for their rare diseases. And so that level of data is actually very easy to share. So working with IRBs and internal privacy offices, we built a website that is publicly viewable, and it basically says these are the number of patients we have consented with each one of the disorders, and then with detailed information of these are how many of these outcome measures we have, this is how we've been watching this. And so it's that very high level information, which is what our PAGs wanted. Well, we were getting bogged down by the details of, well, I can't share that individual level data, but once we actually talked to our PAGs, that's not what they asked for. They needed high
That's right.
level, and high level can be shared, right? And so it's listening, but also being aware of your blind spots of how you're assuming. And so it takes us a while, but we're doing one by one, adding in new disorders. And we also opened up an opportunity where PAGs can come to us and submit a very high level request of, I need this information for X reason. And so it's high level information you can share with a PAG, but it takes resources from us, but it allows the PAGs to come to us and say, I need to know how many kids you consented within X number of months, or which outcome measures you're collecting. And so then we can take that information back to our advocacy partners and deliver them a fit for purpose output. So rather than trying to meet all needs for everything all at once, it's allowing us to be a little bit smarter. Melanie,
I appreciate
you want to jump in?
Yes, no, I appreciate that so much. And I think you're absolutely right. Sometimes it really is just the high level data, and that's all the groups needed. Sometimes groups are asking for information and they don't understand the big picture as well. And so just the honest conversation saying, we can't share this data right now because we don't have the correct consents and we're working to get them, or we're holding this data for a publication that we expect to come out in 6 months. We'll let you know if it's delayed, or this information is proprietary right now and sharing it could jeopardize a relationship with an industry partner, and we don't want to do that. So is it okay if we table this? All of those conversations, I think, have been very well received. They're just not always given the answer right away. And so I think that alone has been really helpful, is when the researchers and clinicians can just share back why. What is the delay? And if the delay is, or the non-sharing is, oh, well, we don't do it that way, then that leads to the next conversation of, okay, what can we change to make sure that we can start doing it that way? But sometimes it's just letting the groups understand what the obstacles are and that it's not just, being blown off. And I think that can be really helpful. And Laura, you've been amazing at that with our advocacy groups and just being really honest and sharing some of the academic roadblocks or slowing down that that can delay data. In the leukodystrophy community, a lot of times people say like time is myelin or time is brain, and so whenever there's a time delay, it can be really frustrating, and we really appreciate the honest answers of what's causing those delays.
Thank you, Melody. I feel like I had a ringer in the audience.
Nicely done. I would like to ask you, Melody, there was reference to an email that you had sent saying the things that you would need. If you would like to share that with us as well, this is something that we can include as two things people might be asking for or looking for that can help guide other groups that are willing to share data of what a low level kind of request might be that might have that easier to respond to, though it does take a little time. Thank you so much, both of you. Sean, did you have something to add?
Yeah, just to kind of follow Laura's explanation, the website idea is fantastic. We do receive similar requests from patient groups that we work with, and we respond on an email basis. But I think having a more public-facing platform website, it's a fantastic idea. So that's something I'm not going to say I'll steal, but certainly take inspiration from if that's okay. But again, going to Teresa, your original question around sort of the requests from individual patients or organizations. It's something that IMPDR are considering at the moment. So I think we can do better in this. And I think part of it is the solution, certainly for the individual patient, is looking at how we can loop them into a potential researcher or clinician who can support them in answering that research question. This is something we've been considering for quite some time. And it's something that we are very keen to pursue. Again, how that looks, I think we have some wiggle room in making sure that we're regulatory compliant with ethical standards and any of the regulation that we need to follow. But ultimately, this is something that is as Özlem said, this is patient data. This isn't IMPDR's data. This isn't registry data. If this data isn't working for patients, then it's not working as well as it could be or should be. So that's a really interesting point that you raise, Teresa. So it's something we certainly are going to take back and look at how we can really support that kind of request.
Thank you.
Excellent. This was a great set of conversations here. That's a great way to start that off. I'm going to now go to Christine Wagner's question. Christine Wagner is our Cure GM1 patient advocacy leader. And Christine, do you want to ask directly?
Oh, sure. Okay. Sorry, my camera's off. I was wondering how these efforts are funded. Are they funded through NIH grants or the advocacy groups? Because honestly, for our own data sharing efforts, it has been difficult to get funding.
I'm happy to start, if that's okay. So our funding streams are quite diverse at IMPDR. So initially, back in 2012, we were funded through a European Commission grant, which was to the tune of roughly 2 million euro. At the time, that grant funding period came to an end in 2016, and our sustainability since then has been grants from non-profit organizations, grants from industry partners in respect to educational grant funding, and also more recently we've entered some commercial contracts through our commercial arm, IMPDR Gateway, which is a separate organization but allows us to work with industry partners in a compliant manner. So really there's a few different streams of funding that we have coming through the registry. But certainly in terms of the international footprint of where we work, we're looking at partnering with institutes in the United States. It may be we look at NIHR working with institutions in the Middle East. There may be different funding streams there. So it's very much being able to identify appropriate funding streams and seeing how your organization meets the funding criteria. So it is quite a, it's almost like a full-time role in itself, trying to identify these grant opportunities. And of course, then coming to the grant application, grant writing can take some time. putting together a successful bid is very time intensive, needs a truly collaborative approach. So it is quite a big ask to undertake, but of course it feeds into the sustainability of the organization and the data collection model, which of course feeds into the data sharing element and the longevity and usefulness of the data collected.
Anyone else want to jump in on funding?
Yeah, I'd be happy to. Hi, Christine. It's nice to speak with you again. I mean, I think the answer is if you're doing multi-stakeholder research, you need multi-stakeholders to pay into the system, right? And so you build a sustainable model by really leveraging all the data sources as well as all of the different funding sources. So we have academic grants, institutional support, strong NIH support, industry support, and patient advocacy group support. And so that allows, as different interests flex in and out, you can maintain a central core research, right? And that goes into the sustainability. The sustainability of a project comes in making a flexible system that can use different support systems in order to maintain itself. I think it's really important also, Christine, it's hard to get funding for a natural history study. Here for that. Natural history studies are really important. It's the mainstay of what I do. They're not very sexy. They don't sell very well. And so I think it's also coming up ways you can leverage the natural history study to do sexier or more enticing work. And so some of that is just a packaging and marketing thing, right? So stratification and prognosis of outcomes, really interesting. Natural history study, a little bit less so, right? And so you're doing the same work, but the natural history study becomes the platform by which you can market an idea or a task. Designing clinical trial inclusion/exclusion criteria using a natural history study platform is one way to make something a little less exciting, more sellable almost to funding agencies and funding groups. And so I think there are ways to create a sustainable large platform across our various consortiums, but selling to funding agencies little pieces of it rather than the whole piece, which again gets to having multiple funding streams to be able to support your work going forward.
Thank you.
Did you want to speak to funding? I know you mentioned it a little bit before.
Oh, it's every single actually project is attached to a grant, honestly. So basically that's how we get the funding. And then the biorepository is kept with our own efforts. So basically it is pretty straightforward.
Great. So we have a couple questions from an anonymous attendee, so I will speak for them. But
That's me,
let's start.
Tara.
Oh, that's love it. So Teresa has another question about GUIDs. You want to talk about GUIDs, Sean?
Yeah, so Sean had touched on it briefly. I just maybe have a conversation offline if you could just give some information more about GUIDs.
Absolutely, Teresa, not a problem. So this is something that we recently developed, and recently 2-3 months ago. And this was a call from the Niemann-Pick community, and this was put together through funding from the Arpa Sagan Medical Research Foundation, who's a non-profit group working in the Niemann-Pick disease type C space. So the ask was to develop a global unique identifier model for Niemann-Pick diseases. So the GUID tool that we have developed, it's a web-based widget, which means it's an interface that sits on our website, impdr.org. And when a researcher uses the GUID tool, no data that they enter is held on a server. It's all generated on the front end. So we don't have any data privacy issues by having data saved on the registry website itself. The GUID uses 4 variables to generate the GUID ID. It uses the family name of the patient, the first name of the patient, the sex of the patient, and the patient's date of birth. So using these 4 parameters, we use some cryptographical science and magic, which I don't completely understand, but basically it's a way of reforming those four variables and producing a string of text that is non-reversible. So you couldn't take the GUID string and then work out the name, the date of birth, and sex of the patient. So the vision is you would have a patient within 2 data sets, and you'd have those 4 parameters available, and you'd have a researcher enter those variables into the registry website, and it would produce this GUID ID. Then through a conversation or some other approach, the researcher with GUIDs in this data set will be able to find the GUIDs of the researcher's data set over here and link the two. So the technical solution is freely available. It's on the IPDR website, but the governance solution, and in terms of the actual operational side of things of matching those records, that will be very much driven by the individual researcher themselves. So they would need to go away, look at their ethics approval that they have in place to see if they could facilitate this linkage of the data. They would need to look at their consent forms to see if they could get, or if they have approval from the patient to undertake this GUID process. And then they would need to be able to link up with researchers with other data sets to have this conversation to see how they could match these GUIDs. So that step in terms of the operational side of things and the governance side of things, that sits very much with the researcher. We can have conversations with those researchers who are interested in using the GUID. And in fact, on the website, we do have a primer, an FAQ of how to use the GUID, and it goes into the technical and the operational side of things. And of course, we can always have a conversation with those researchers. But as far as we understand, this is a novel approach, not just in Niemann-Pick diseases, but also in the LSD space. But it is something that the wider community are very interested in producing and facilitating in their own quarters. So it's something that we'll be talking about more in the coming weeks and months. It's certainly something that we're going to be leading with this year when we're speaking at conferences, and we're looking to get a few publications out of this because, again, dissemination of this process and how we came to this tool, I think, is of big interest to the community. And of course, Teresa, happy to connect offline and have a more individual conversation if that's something you'd be interested in.
Absolutely. Thank you.
I just wanted, I would just wanted to add, you know, working with Sean, as well, the INDPR, as well as our procedure, it also brings back, you know, one of the things we showed in the first webinar, and that was Christine Wagner's roadmap, in which there's different barriers. And this, you know, just to be completely transparent, this is also one of those barriers, in that while the INDPR has created this international GUID ID, there's still, and there's an NIH GUID toolkit, getting the researchers, now they would have to go back in and insert in duplicate these forms. And yeah, these cells, yeah, it's only 4, but you know, we're also talking about researchers that have a very you know, high load in terms of patient, and so it still is a challenge. It's not, this is a potential solution, but it's not without its challenges. And you know, it just goes back to that visual that Christine said. There's always seems to be, oh, we find a solution and then we make problems out of the solution. And it's a continuous give and take, and you know, it is you know, we're moving towards that. And I love the fact that the INDPR has the international GUID, but just to be clear, just creating that doesn't solve all the problems. It's just the first large step, but there's a lot of steps after it. That's, I just wanted to add.
Thank you, Krista. That was part of the issue that I had a question about, because I know that the NIH has a system, and then there are other systems. So how do those two systems talk to each other? So that was very helpful. Thank you.
I just want to unfortunately wrap us up right now, because just as the conversations start to get really exciting. But I think I would just like to thank everybody that has presented today, Azam, Laura, and Sean, and my co-presenter, Krista. Also, I would like to thank, I did not do this last time, our wonderful support staff from both the Lysosomal Disease Group at CPATH, as well as the Orphan Disease Center. Nicole, Olivia, and Leslie have done so much for both of us, and we're so appreciative for everything that they've done, everything on the website, all of our technical aspects, it is all them. So thank you so much for making this work as well as it has. I just want to go to a quick summary slide here that encapsulates some of the stuff that we've seen so far today. It's not thorough, but it definitely has a lot going on. And this is really just as a quick snapshot of what we've accomplished so far today, but there's so much more for us to discuss here. So we're really excited to see that next step for the work that we are doing here to make sure that data can be shared well, efficiently, with privacy, and to advance our science in all the ways that we've been talking about today. So I'm not going to go through all of this, but those are some of our highlights from our presenters today. And I just want to say another plug for our next 2 sessions coming up. Case study number 2 for retroactive data sharing. So how you would approach data sharing once the study has already begun or has closed. And then finally, we will wrap it up in June with data sharing collaboratives. You will see a repeat of some of the people that you've already heard from so far. And of course, we always need more case studies needed. So please give us some case study ideas, work with us to either submit as a presenter in real time or to have your case study added to the literature that is created later. We'll take all feedback that you may have. Thank you so much for everybody for your time, and we will see you soon. So see you next month. Thank you so much, everybody. Have a good one.
Thank you.